Hello, everyone. So it's a great pleasure for me to introduce today's speaker at Global IMMUNOTALKs, and he's Dipyaman Ganguly. So Dipyaman is a professor and the chair of the Department of Biology at Ashoka University in India. He studied medicine in the Medical College of Kolkata in India, and then he moved to the US to do his PhD in immunology. So this was in the lab of Michel Gillier at the MD Anderson Cancer Center in Texas. And when he was there, he worked on the activation of dendritic cells by complexes made of antimicrobial peptides and self-RNA, which can trigger autoimmune reactions. So from Texas, he moved to New York City to the lab of Boris Reisis at Columbia University. And he remained interested there in the link between dendritic cells and autoimmune diseases, starting working on the role of plasmacytoid dendritic cells in systemic lupus. So in 2013, Dipyaman completed his training and he started his lab in India, and his lab focuses on human immunology. So he keeps studying the role of PDCs, plasmacytoid dendritic cells, in autoreactive inflammation and the underlying cell biological mechanism. But he also developed another research project on the key role of mechanotransduction, in particular through the Piezo1 calcium channel in human T cells. And I believe this will be the subject of his talk today. So Dipyaman was recently elected fellow of the Indian National Academy of Sciences, as well as of the Academy of Medical Sciences. And he's a member of the Royal Society in Biology in the UK. So my special question for Dipyaman today will be, in what ways has interdisciplinarity and collaborations enrich your scientific perspective? Please, Dipyaman.
Hi. First, let me tell, so it's a great privilege to be able to share our work in this really prestigious platform. Thanks to you and all the global immune organizers. Now, coming to the question, I have always been surrounded by collaborators, and I never left any stone unturned to garner all the support that experts from other areas can give us in terms of answering our questions. So in the talk itself, you'll find that we closely collaborate with physical biologists as well as mouse geneticists. So we are on human immunology. We are focused on human immunology, but we have close collaborations with others. I think the collaborations can actually take you to places, and always it's always welcome. I mean, better to welcome collaborations so that the questions you are answering can actually be answered fully. I'll stop at that.
Okay, thank you, Dipyaman. And I hope the young people will follow your advice and think of collaborating, even sometimes with people that do very different things. And now you can share your screen and go ahead. The stage is yours.
Okay, thank you. So let me share.
It's great. You can go.
Okay. Yeah. So as Marie already introduced that, I'll be talking about Piezo1 mechanosensing in human immunocellular functions. I'm from Ashoka University in India. And so today my plan is to introduce briefly the major questions that we have been interested in our lab, and also how these questions led us to newer questions and took us to places, and how we arrived at looking into Piezo1 mechanosensors and what got us interested in that. And then I'll briefly talk about two apparently discrete projects, but both of them linked to really fundamental functional dimensions of human immune cells, where mechanical cues play a very important role. Now, before going to the data, so all the research that I'll be presenting today are done by this brilliant group of graduate students. A lot of them have contributed to whatever I'm going to talk about today. And for example, Deblina, Chinky, Purvita, and Shrestha. So I'll actually touch upon work done by them today. And we have been reasonably supported by funding agencies, both public and private in India. Now, our major interest has been in this dendritic cell subset called plasmacytoid dendritic cells, which I'll keep on referring to as PDCs. Now, these are a lot of them share ontogenic origin with conventional dendritic cells. That's why they're called DCs. But they hardly look like dendritic cells. They don't have dendritic processes. They actually look very much like lymphocytes. And they do present antigen, but the antigen presentation is not their major function. They're more known for producing type 1 interferons. And they have a very intricate secretory phenotype. Under electron microscope, you find really intricate Golgi bodies. And that's because of its secretory phenotype, because it can produce loads of interferons in response to toll-like receptor activation and very quickly. And there is the other cell that has this robust secretory phenotype among immune cells or the plasma cells, and that's how they derive their name as plasmacytoid dendritic cells. I think my headings are not visible. Anyway, I'll just go ahead. Now, as I already mentioned, the major function is producing type 1 interferons, and these cells produce type 1 interferons in response to toll-like receptor activation. They have endosomal toll-like receptors 7 and TLR9, and these toll-like receptors are equipped to recognize nucleic acid ligands. And when you're talking about nucleic acid ligands, they can come from both pathogens as well as from the host. And so pathogen origin nucleic acid ligands are recognized by these cells. They produce type 1 interferons, and they are really major responders to especially viral infections. Now, because self-nucleic acids could also be recognized by these toll-like receptors, they are also implicated in a lot of autoreactive inflammation. It has been shown that genomic DNA, mitochondrial DNA, free RNA, when they're in complex with endogenous proteins or autoantibodies, they can actually get access to the endosomal compartments, and then they get recognized and the cells are activated. Now, as I said, the nucleic acid recognition, nucleic acid of self-origin recognition leads to a role played by PDCs in chronic autoreactive inflammatory contexts. Almost 2 decades back, we started working on looking at role of PDCs in the autoimmune diseases with Michelle in MD Anderson and also with Boris in Colombia. And what we found that nucleic acids released by dying cells in different tissues. It can be in the skin or neutrophils which are circulating in the blood. They undergo a death called netosis. And these nucleic acids can be recognized by PDCs, and that leads to interferon production, which then lead to a lot of secondary immunocellular phenomena like T cell proliferation, better presentation of antigen to T cells by conventional DCs, B cell expansion, autoantibody producing plasma cell expansion, etc. And thus they are implicated in different autoimmune contexts. During our work, we had implicated them in psoriasis as well as in systemic lupus. And so till then, it has been shown that especially in systemic lupus, PDCs play a really crucial role. And because of this work and also other groups like Jacques Moschro's group and Virginia Pascual's group have established a critical role of PDCs and interferons in SLE that led to the monoclonal antibody development like anifrolumab, which is a type 1 interferon receptor blocker and is approved for clinical use in high-grade lupus nephritis, as well as there are monoclonal antibodies developed against PDCs called litifilimab, which are undergoing clinical trial right now. Now, when we are talking about autoimmune diseases and role of type 1 interferons from PDCs in these diseases, there were also clinical data showing indications that type 1 interferon abundance systemically may also have to do something with systemic insulin resistance, because there is, like in the autoimmune diseases, there have been patients who are coming back to clinic with metabolic inflictions, for example, very commonly insulin resistance in psoriasis patients, cardiovascular complications in rheumatoid arthritis patients, etc. And also the pegylated interferon therapy, which used to be used for viral infections, that also led to insulin resistance development in a lot of patients. And that led us to investigate this. And we actually looked into obese individuals, trying to find out whether PDCs and interferon production by PDCs play a role in the metaflammation in visceral adipose tissue. As you know, that in the obese individuals, there is a pro-inflammatory milieu in the adipose tissue, and we first showed that PDCs do infiltrate in the adipose tissue. They recognize self-nucleic acids released by the dying adipocytes, produce type 1 interferons, and that actually drives a lot of secondary immunocellular phenomena, for example, depletion of Tregs or macrophage polarization. So our study was done on human samples. It was pure human immunology work, but since then it has been validated by different genetic models. For example, TLR9 knockouts are protected from high-fat diet-induced type 2 diabetes. PDC-less mouse, both E22 knockout and BDCA2 DTR mice, they have been shown to be protected from insulin resistance in response to high-fat diet. So we actually implicated that self-nucleic acid recognition is driving PDC activation in adipose tissue. And recently we have teamed up with Vania Sisirak in Bordeaux. So Purbita Bandyopadhyay was a graduate student in my lab. So Purbita first showed that DNS1L3 is a nuclease that cleaves free nucleic acids and also in the apoptotic bodies. And we found that DNS1L3 was depleted in some of the obese individuals. And that data was validated by Vania in the DNS1L3 knockout mice, where we see that in the knockout mice, in response to high-fat diet, the insulin resistance develops much faster and to much aggressive extent. And so you can clearly see that this event of self-nucleic acid recognition by PDCs and production of type 1 interferons are implicated in different diseases. And other groups have shown that this is also implicated in systemic sclerosis, Sjogren's syndrome, type 1 diabetes, rheumatoid arthritis, etc. And that becomes a central innate immune activation event in all these diseases. Naturally, it also becomes a very interesting therapeutic target. And so we have been part of wider efforts to targeting PDCs with small molecules, mostly driven by others, medicinal chemist groups in India. And we could develop certain endosomal toll-like receptor antagonists in collaboration with these bigger groups on medicinal chemistry. Now, while we were working on these PDCs, some of the students in the lab were really interested in exploring the cell biology and regulation of PDCs, the human PDCs. So we work on them by collecting human PDCs from the circulating blood of healthy donors, and then we work on primary plasmacytoid DCs. And we have been interested in the extrinsic regulation, what are the soluble mediators that can actually affect PDC activation, and also endosomal pH regulation, endosomal trafficking. And that's really important because endosomal trafficking, it is presumed that endolysosomal trafficking needs to be tightly regulated in PDCs, because all these endosomal TLRs, they reach the endolysosomal compartment and then they get active at a specific pH. When the proteases that are active in that pH cleaves off a part of it to make them active. And naturally, the endolysosomal trafficking as well as pH regulation is really tightly regulated in this sense. And it's important for their function. Now, I'll focus on endosomal trafficking for today's work. And while we were exploring that, we caught up in this physical cue hypothesis. And so this is known in the field for a long time, that there are oligonucleotides, mostly CpG-rich oligonucleotides, which are bona fide ligands for TLR9. And in the field, it was known for a long time that different oligonucleotides, different CpG oligonucleotides, like A-type oligonucleotide, which I'll mention as CpG-A, or the B-type oligonucleotides, I'll mention CpG-B. So they actually lead to different functional outcomes. And also it was reported that some of these oligonucleotides, for example, CpG-A has higher aggregation property. And people never could link it through until almost 20 years back, when Professor Taniguchi's lab showed in a very interesting paper, they showed that these two oligonucleotides are actually dealt by the PDCs in different ways. For example, they showed that CpG-A, they largely interact with TLR9 from the early endosomal compartment, and that leads to predominant IRF7 translocation to nucleus and production of type 1 interferons. On the other hand, CpG-B was known to be not very efficient in driving type 1 interferon response in PDCs. And Tata's group showed that CpG-B oligonucleotides quickly moves to the late endosomal lysosomal compartment, and they fail to induce enough IRF7 translocation, and their other drive NF-kappa B activation, and that leads to pro-inflammatory cytokine production, but not type 1 interferon production. Now, it's very interesting, because now you can see the idea is that the cargo size or structure, because the CpG-A oligonucleotides are known to form aggregates, while the CpG-B oligos are usually monomers. And that tells you that this difference is being perceived by the cell in different ways, and they're actually cells are responding to them differently, so that the functional outcomes are different, and there must be cell biological regulations how this is happening. And we were really intrigued by this. Actually, 3 graduate students worked on this work, so that it was started by Devlina and then continued by Purvita and Shrestha. I'll actually show data that all 3 of them have generated. Now, first, we wanted to see the CpG-A and CpG-B that we are using, so whether they are having this telltale sign of aggregation properties, etc. So we actually did single angle X-ray scattering experiments. So you can actually see the minimal units of CpG-A are really much bigger than CpG-B. And when you also do fluorescence correlation spectroscopy, so these are done by Ashish Ganguly in IMTech India and Krishnananda Chattopadhyay in IICB India. So they're really experts in these fields. And so what Krishnananda showed that yes, even in the fluorescence correlation spectroscopy, CpG-A has a much higher propensity to form aggregates, while CpG-B hardly does that. That was confirming our oligos that we are using. And then the question is, why is this difference of functional outcome? Is it because of they're using different endocytic pathways? And they've been actually addressed that. And we used inhibitors and also the driver adapter molecule for click-GIG pathway. So we actually targeted clathrin-dependent endocytosis, caveolin-dependent endocytosis, and click-GIG pathway, which is clathrin and caveolin independent. And we found that there is not much difference between these 2 oligonucleotides in terms of utilization of the endocytic pathway. So both of them were going through click-GIG pathway in these human primary PDCs. And then we did one scanning electron micrograph, and that was interesting. So Devlina looked at these cells after they have been treated with these oligonucleotides, and she found that within 10 minutes of adding these oligonucleotides on the cell culture, the PDCs which have seen CpG-B, they don't really change much. But when you are giving CpG-A, they show a lot of membrane rufflings on the surface, and that was interesting. And so we kind of hypothesized, perhaps because of the bigger cargo, there are so much of membrane rufflings that's happening on the cell surface. And so we hypothesized that maybe that because when you are coming with the CpG-B, which is a really smaller cargo, it's not really making a lot of, I mean, the size of the endocytic pit is much smaller, and that's not creating a lot of membrane stretch. On the other hand, and there is not much physical cue. But on the other hand, when you are coming with a bigger cargo, because essentially we are looking at difference because of the size of the cargo. And so when you are coming with a bigger cargo, for example, CpGA, it causes far more membrane buckling, and that perhaps creates membrane stretch and some mechanical cue, and perhaps that's how the functional outcomes are differentiated. Well, this was a outrageous hypothesis, but we had to first see whether there is indeed a change in the membrane tension. And that's why we needed collaboration with Dr. Vidisha Sinha, who is in IISER Kolkata. She's a physicist turned cell biologist. And she used a microscopy method called interference reflection microscopy, which is essentially, so because they have shown that when you are I mean, on a resting state, on a steady state, cell surface is really fluctuating, which is energy dependent process. And when there is membrane stretch on the plasma membrane of the cells, these fluctuations dissipate and they come closer to the cover slip when you are doing microscopy. And so in this microscopic method, you can actually look at the interference that these two light beams are experiencing and measure how far the membrane is from the cover slip. And that can be related to the membrane tension that the plasma membrane is experiencing. And that's a very nice way of, I mean, without intervention, without any physical intervention, you can actually measure membrane tension. And we actually employed this technique for looking at our PDCs. So when we treated our PDCs with CpGB, you can actually see it's a nice normal distribution of membrane tension. We are actually looking at the membrane tension. And when you give CpGA, again, it follows the same tension profile, but there are outlier regions on the cell which show higher tension. That was interesting. So that we hypothesized maybe because of, so these are the areas where CpGA is entering the cell. But to confirm that, we did a TARF as well as IRM together, where you can actually look at the fluorescent ligands getting into the cell, being attached to the cell surface, and then do the interference reflection microscopy around that puncta and try to look at how the local tension area is changing. And you can actually see, so you're looking at cells. So these green puncta are the labeled CpGA, and these are the IRM maps. And here we look at the normalized tension map at the distance from the center of the bright puncta, right? And so we did that at different time points, 5 minutes after addition of the oligos, 15 minutes after addition of the oligos, and 20 minutes. And so you can actually see, this is a baseline, the black dot, black line. But if you look at the pink line, which is the 5 minutes after addition of the oligos, there is little increase in the tension, but not much. But within 15 minutes, it goes really up. So that means there is a high tension ring forming around the cargo, which is fluorescently tagged. And so that made us more comfortable that with the idea that, yes, there are membrane tension generation happening around the cargo near the, around the endocytic pit. Then another question is, if this is the only cue that the cell is having to have functional diversification, how is it getting sensed? There need to be a sensor that will actually sense it. And that brought us to Piezo1 channels. Piezo1 is a mechanosensory calcium channel, as you know, and this is a really professional mechanosensor. It doesn't really need other proteins to sense membrane tension on the plasma membrane. And it has a very intricate trimeric form. So these blades juts outside the membrane. And when there is horizontal tension on the plasma membrane, the blades come down and the central pore opens, which allows a bivalent cation, mostly calcium, to go in. And that's how it senses the membrane stretch. And so there have been a lot of studies on this Piezo1 channels, and Piezo1 is more ubiquitous compared to Piezo2, which is largely restricted to the central nervous system. But for Piezo1, the study that attracted our attention back in 2015 was that Piezo1 is really important in erythrocyte volume regulation. That told us that it's expressed in the hematopoietic system. And then we went into all the public datasets and found that yes, it's actually expressed in almost all immune cells, more so on dendritic cells, macrophages, and T cells. So first experiment that we did, we wanted to see what's happening with Piezo. So we are staining for Piezo protein in the PDCs. This is an untreated cell, and this is the cell which is treated with CpG-B oligonucleotide, and you're looking at Piezo protein expression, and you are coming back with CpG-A. Now, within 10 minutes, you can actually see there is a really bright bright Piezo1 puncta on the surface of the cell. And that was interesting. So that told us that perhaps Piezo1 is getting engaged when you are actually giving these oligonucleotides. And then we did some functional studies and used RNA interference, because we are working with primary PDCs. So we used RNA interference to knock down Piezo1 expression on these cells. And so, as I told you, CpG-A is more efficient in producing type 1 interferons, CpG-B is not. Now, if you knock down Piezo in these cells, CpG-A-induced interferon induction also goes down. Now, if you come back with CpG-B on the PDCs and add one agonist for Piezo1, which is IODA1, and then again, CpG-B can now produce a lot of type 1 interferons. Now, this induction of type 1 interferon in response to CpG-B, when you're you're using an agonist for Piezo1, again, is absent when you're knocking down Piezo. So that was showing that Piezo1 has a role to play in differentiating these two oligonucleotides. And then came the question of this differential transcription factor translocation. So as I mentioned, that CpG-A is known to drive more IRF7 translocation. And within 30 minutes, you can actually see IRF7 translocated into the nucleus. And GSMTX is a peptide inhibitor of Piezo1. And when you add GSMTX and then come with CpG-A on the PDCs, you see not much IRF7 translocation is happening. Instead, NF-kappa B activation is happening. And so that kind of established that, okay, so Piezo1 activation was linked to this differential functional outcomes. Now, the question is, how Piezo1 activation and endosomal traffic linked? And that was a very difficult question to answer. And we have been looking for clues. Now, meanwhile, while we are working on this PDC problem, a brilliant graduate student in the lab, she got really interested with Piezo1, she's Chinky Shu Chen Liu, and she said, okay, so you are working with PDCs. Let me look at more fundamental things in another cell, for example, T cells. What was the fundamental thing? So we know that T cells are interacting with DCs and antigen presentation is happening. And we usually mimic them in vitro. All the immunologists have been mimicking it by using cross-linking antibodies to CD3, CD28 so that the TCR complex is activated and T cells are activated in vitro. Now, all of us also know that these cross-linking antibodies have to be usually bound to the plastic plate for having optimal T cell activation. And these days, you usually use a solid matrix bead where the cross-linking autoantibodies are, cross-linking antibodies are actually bound. And so that told us that maybe there is some physical cue needed so that the cross-linking antibodies need to be on a solid matrix, and the cells should bind the cross-linking antibodies on that solid matrix so that the physical cue is intact. And so Chinky actually showed that yes, Piezo1 is expressed, as I already mentioned, on human T cells, and she showed that this solid matrix bound cross-linking antibodies to CD3 and CD28 could not activate T cells optimally when you knock down Piezo1. More interestingly, because you are doing knockdown on human primary cells, so different donors will actually, different experiments will actually have different levels of knockdown. And that can be used to validate the relationship with the functional outcomes. And here you are looking at a graph where you are looking at the percent knockdown of Piezo1 for a specific experiment. And then you're looking at the first signaling event after T cell activation, which is phosphorylation of ZAP70, and you can actually see the correlation. So that was interesting. I'm not going to go into detail, but what Chinky also showed that Piezo is known to induce calcium influx, and it does that in the activated T cells as well. And this calcium then activates calpain, and which is then linked to cortical cytoskeleton, actin cytoskeleton bolster. And she showed that when T cells are interacting with this peptide MSC complex or the cross-linking autoantibodies on the solid matrix beads, there are membrane tension happening, and there is piezo activation. Calcium goes in, that activates calpain and does this effect in, drives this effect in polymerization, which bolsters the cortical cytoskeleton on which the immunological synapse is perhaps sitting. And perhaps that's how the T cell, optimal activation of T cell is ensured. And it actually became the first evidence of a crucial role of Piezo1 in any immune cells. And then Chinky took up a more complex problem of T cell migration. And although the problem was kind of simplified because we were actually looking at 2D migration on an ICAM bed. And so during migration, what happens, for these amoeboid movements, there is a leading edge that is actin polymerization. This actin then flows backward towards the rear edge. And with the help from myosin interactions, it generates force that propels the cell forward. And whatever is known in the field is, in response to a chemokine, when T cells are moving, when the chemokine receptor gets activated, that activates focal adhesion kinase, and focal adhesions are formed. And then that is followed by integrin activation, which again gets recruited to the leading edge, that drives some of the effect in polymerization. And this effect in polymerization is further amplified, and the retrograde actin flow starts. But it was not very well known how focal adhesion kinase is actually linked to the integrin recruitment. So what Chinky did, a very simple experiment, she did a 2D chemotactic assay with the human T cells, and we did RNA interference of Piezo1. You can actually see there's a control siRNA. T cells are moving nicely towards the chemokine gradient. When you are giving Piezo1 siRNA, they sort of lose the way. So their polarity is kind of lost, and which can be also looked at through this analysis. And when we did a live imaging on Jurkat cell, and we expressed fluorescent Piezo1, and we found that Piezo1 reorients itself towards the direction of the movement. And that was really interesting. And then we again went back to Vidisha and tried to look at whether there is membrane tension forming. And she actually found, using the same interference reflection microscopy technique, that membrane tension generation is followed by Piezo1 recruitment towards that membrane area. And that was, again, supporting that the Piezo1 recruitment at the leading edge and sensing of the mechanical cue was really important. First, we asked whether the focal adhesion kinase formation is dependent on Piezo1. We found no, it is not. On the other hand, Piezo1 recruitment to the leading edge was actually dependent on focal adhesion kinase formation. And so also the rear edge actin accumulation was dependent on Piezo1. And so you can actually see here a moving cell. So the yellow signal is coming from Piezo1, and it's in the leading edge. You can actually see the lamellopodia forming, and there is a retrograde actin flow which forms the uropod and propels the cell forward. So what Chinky showed that Piezo1 is kind of linking these two events, focal adhesion formation and the integrin recruitment and the retrograde actin flow. Now, so that was fantastic, although in the T cell field, role of Piezo1, there has been really contradictory data from different contexts. For example, it has been shown none other than Ardem Pataputian, long back 2018, that gain of function Piezo1 in T cells protect mice from cerebral malaria. On the other hand, other groups have shown that the effect in the genetic models are at best modest. People have shown that Piezo1 channels restrain regulatory T cells, but are dispensable for effector CD4 T cell responses. On the other hand, fluid shear stress has been shown to activate T cell activation through Piezo1. Then other studies in the tumor model have shown that Piezo1 facilitates optimal T cell activation during tumor challenge, etc. So the verdict is not out yet. Now, in our case, we are actually looking at a 2D migration, chemotactic migration model. Now, what Chinky's studies gave us is a clue that, okay, Piezo1 now is actually linked to effect in polymerization in the local area where Piezo1 activation is happening. And so let us come back to our original question in PDCs. So we were not able to figure out how the Piezo1 activation is linked to the early endosomal retention of the bigger cargoes, right? And how is it linked to the endolysosomal trafficking? And so then we thought maybe, so because we already talked about this, the hypothesis is that a smaller cargo will have less mechanical Q on the surface, a bigger cargo will give a higher mechanical Q, and that will activate Piezo1. There will be calcium influx. And we found that, yes, when you give CPGA, the aggregate forming oligonucleotides on PDCs, there is calcium influx. And when you knock down Piezo1, this calcium influx is reduced or inhibited. And there is evidence for calpain activation. And I don't show all the data here. And we hypothesize that perhaps calpain is driving a local effect in polymerization around the endosomes, early endosomes, and which is kind of retarding the maturation of the early endosome or their fusion with the lysosome, etc. And now this has been shown previously, how the effect in comets and effect in meshwork can actually regulate the transport of vesicles on the microtubules. It has been shown in synaptic vesicles that local actin meshwork can actually retard synaptic vesicle from going to the junction. And also effectin comets are really important for driving the endolysosomal traffic. And if there is a lot of actin polymerization around, and there can be a retardation of its movement. So we wanted to answer this through microscopy. Again, we are actually looking at actin and the two markers of early endosome and the late endosome. And we are getting rid of the cortical actin and only looking at actin, which is not part of the cortical cytoskeleton, and then trying to look at how closely they're forming with respect to the early endosome or the late endosome. You can actually see, so CpGA, the enrichment of actin with early endosomal compartment. When you inhibit Piezo1 using this peptide called GSMTX, the actin enrichment around early endosome reduces. And so that was supporting our hypothesis that perhaps this actin meshwork is retarding early endosomal maturation. And so the cargo is getting retained in the early endosome for a longer period of time, driving IRF7 translocation. So I'll just stop here. And so these are the ongoing and future studies. Some of the studies we have already started. For example, it's already been shown by other groups that Piezo1 is playing a role in the phagocytic functions in innate immune cells, especially macrophages, in the context of pathogen entry. For example, these two work showed that bacteria phagocytosis needs Piezo1 activation. We are also trying to, being a human immunology lab, we think that this Piezo1 response in different immune cells, and there are human polymorphisms of Piezo1 which do affect Piezo1 function in different ways. They are not loss of function mutations, but they change the Piezo1 dynamics and also the kinetics of the calcium influx, etc. And that can actually subtly change Piezo1 function. And we want to look at whether that has any effect on the heterogeneity of immune response in terms of how different immune cells, their functional outcomes can be differing in relation to the polymorphisms of Piezo1. And definitely we are collaborating with other groups for genetic validation of the Piezo1 mechanosensing in immune cells, but our special focus is on dendritic cell subsets. I'll end here. I'll be happy to answer some questions.
So thank you so much, Dipyaman, for this beautiful interdisciplinary work on fascinating Piezo1 mechanosensitive channel. So I will remind to all those people watching your talk that they can ask the question on social media, the Blue Sky Network in particular. And yes, I wish you a nice end of the day, and see you soon all in Global IMMUNOTALK. Bye-bye.
Thank you so much. Thanks, Zara.